So yeah, I'm not Surma Bhopali. Yeah, but I'm Snigdha Thakur. uh, though I come from Bhopal, and we are at ISA Bhopal Department of Physics. Let me start thanking the organizers, Mahesh and Ranjini, for organizing such a wonderful talk for all the students, though I could not attend most of it. I am getting feedbacks from the students sitting here, and they are pretty happy about the way it has been organized and the talks that are going on. So thank you. Thanks a lot. And thank you for giving me this opportunity to share our recent work with the brilliant young minds sitting here. And I would like to get feedbacks also from all of you. Feel free to interrupt during the talk, and no hesitation, even if it's a silly question, please ask those questions. I would be really happy to entertain those questions as much as I can. Okay. Like Samjit said yesterday, most of the part is going to be soft. Though I will motivate you towards the end that yes, these models that we are developing can be applied for the living systems also with some analogy. But yeah, it will be mostly soft is what I'm going to talk to you about. Okay. So as you see, the title says dynamics of chemically active flexible polymer. Okay, I'm sorry for this thing. There are two actives which I recently changed just today morning. That's why two active has come. It's chemically active polymers, right? So before going to the active polymer side, I would like to tell you a bit about active matter. I am not sure how many talks have happened, and if the talks have happened on active matter, you must have seen these videos where people talk about systems which are out of equilibrium by virtue of a constant energy flux which is given to them, right? And the origin for these energy flux could be very different. There is a range through which the energy flux can be given to the system and which drives the system out of equilibrium. But the underlying line is that these are systems which are out of equilibrium by virtue of some energy which is being provided, which it is generated by itself most of the time by some mechanism, right? And these active systems are there at all the length scales that we can think of, starting from nanometer to all the way till meter scale, our length scales and all that. So if I go to very small length scale, the familiar example that everybody gives is of bacterial system. And you can see that there are different types of bacteria. Okay, the first movie you see there are bacteria which have the flagella. The second bacteria is the one which doesn't have the flagella. But the bottom line is all of them move. Okay, with some, so what is obvious to us is that bacteria with flagella will move with a certain different mechanism, and bacteria without flagella will move with a certain different mechanism of propulsion. But the idea is that all of them move, all of them have some energy source with them which makes them move, right? And in nature, we find these motors, these active systems at various length scales. Bacteria, the motors that we have in the body is one extreme. The other extremes are fishes, birds, humans, all of us are different extreme of the length scale, right? An interesting thing is that at all these length scales, even though mechanism of propulsion is very different from each other, right? The behavior, especially the collective behavior, is very similar in all these length scales. Okay, so that was about the active systems that are there in nature. And then of course we all the humans are always inquisitive about mimicking the nature, right? And we have been mimicking the nature by making motors at larger length scales, right? We have these cars and bikes and aeroplanes, right? Those are motors at large length scales, right? And that has been realized since long. But recently, about 20 years back, people have started questioning, asking this question that can we synthesize motors which are miniaturized, right? Which are of the length scales of nanometer or micrometer, right? And the idea is that if I can do that, if we can do that, then that can be utilized for different aspects altogether, right? That can be utilized for drug delivery purposes, that can be utilized for chemical sensing and many other stuffs which is required at the small length scale, right? But one thing that needs to be kept in mind is that when we try to miniaturize the system, right, whatever was the mechanism which was working at larger length scale will not work at the small length scale. Why? Because the forces which governs the system at large length scales are completely different. It's mostly inertia that plays a role there. But the moment you come to a very small length scale, inertia vanishes. Not vanishes, but becomes very slow. Right? And therefore, those mechanisms will definitely not work. One has to bring in some other mechanisms to make these particles move and then study them in the collective motion and various other stuff that one can do. Right? And this is one type of particles. Of course, there's a plethora now. Since this work has started 20 years back, there's a plethora of particles which shows active behavior, that means self-propelled motion. I'm just giving you few examples just to get an idea, especially for students who have not come across this area. Right? For example, this is one typical example of synthetic active particle. This is nothing but a micron-sized Janus particle. Right? These particles have two faces. So you can see this is a silica particle which is half coated with platinum, right? And what this platinum half coating does is it converts. So when this Janus particle is inserted in hydrogen peroxide solution, there is a reaction that goes on only on the platinum site of the colloid, right? And because of that, what happens is there is an asymmetry of the chemical, the product which is generated by virtue of this localized reaction happening on these species, and that gradient is sufficient for this particle to move by the phenomena, something called diffusophoresis, right? So phoresis is a phenomena which is known again by 1960s and 70s, where it was shown that if you have a gradient, if you put a particle, a colloidal particle in any gradient, thermal gradient, electrical field gradient, chemical gradient, it is going to show a propulsion, right? Now what is here, what is happening here? This is self-propulsion, it's self-diffusophoresis, where the particle is generating this gradient by itself, so you don't need any external agency, right? There's an agency inside itself which can make it have this self-diffusophoresis, and then it can move by that mechanism, right? And then again, people have tried various, I'm just showing you silica platinum. There's a various kinds of these kind of particles which people have synthesized, studied theoretically, and using simulations and so on and so forth. Okay. There's another class which people have started talking about very recently is the deformable active particles, right? Now, these are all rigid active particles, rigid synthetic active particles. Now, the question came that what will happen if we make them deform? And yesterday you saw you saw the talk from Rajesh, right, where they were talking about deformable active particle. This is one such active particle, which is a vesicle, which is a synthetic vesicle. It's asymmetric polymerisome, right? So what they do is they make this vesicle, which is made up of polymers again, with two different entities on two sides, and there's some reaction that goes on inside this, and this reaction, basically hydrogen peroxide going to hydrogen and water again, and because of this reaction, one side is being more semi-permeable, other side being less permeable. This gradient is again generated along this vesicle, and that propels itself just like the silica platinum particle. Only difference is now this is deformable, therefore the dynamics is going to be much more richer than before, right? Okay. The other class of active particle, I'm sorry, that I would be interested in is active polymer. These will also come under the category of deformable active particles because these are polymers and that can change their shapes with dynamics. The structure and dynamics are correlated, unlike the glassy system. Here the structures and dynamics will be correlated with each other, and that's on the system that we are going to talk about here. So these are nothing but chain-like structures in which the segments exhibit active motion. So these will have segments which will be active, right? One example, some of the examples that I will take you through. One such example is this kind of filament which is made by, again, the same Janus particle, okay, that I was talking about. But these Janus particle, by virtue of some AC electric field which is applied to them, they self-organize into such filaments or the rings, okay, And by self-organizing themselves into these kind of filaments and ring, they show various kinds of motion that you can see here. They can show a free forward motion. They can show a motion where they buckle a lot. They can also show a motion where they can oscillate just like cilia. Okay, so these are the filaments which is made again from those tiny genus particle, but very rich in terms of their dynamics, which is related to the structure of these active particle polymers. Okay, this is one example. Self-assembly, okay, so these are the dielectric particles. You can see one side of this is silica, another side is titanium. These are the dielectric particles, and when you apply the AC electric field, then there is an interaction. So basically electrophoresis that happens, and that electrophoresis aligns them into a certain way. Okay. You can look at this paper for details of the mechanism. They have talked about the details a lot. Okay, the other example. So I'll give some more examples and then we will go to our work. Another example of active chain is where the chain is made up of passive bead itself. These are magnetic colloids, okay, colloids which are having magnetic particles, and then when there's a magnetic field which is applied to this, it aligns into this kind of chain-like structures. However, this magnetic chain is now put in the bacterial solution. Okay. Now, what this bacterial solution does, it imparts the activity, it imparts forces to this chain. Okay, and because of those activities which are imparted by the medium now, okay, it shows a very nice motion, so which is depicted by this mean square displacement, which shows that when you have activity induced, the mean square displacement is pretty high for these chains, as opposed to when there is no activity induced. That means there are no bacteria in the solution, right? So that shows the enhancement of motion by the presence of activity, and in this case the activity is brought by the medium, not by the chain itself, which was the case in the previous slide. They again change the density of the bacteria, basically. In terms of that parameter alpha? It's basically related to the bacterial density. Okay, I exactly don't remember how it was exactly with alpha, but it is related to the bacterial density. So that means more kicks. More is the bacteria, more is the kick that is going to happen. Okay, right. One more example of lab-made active polymer is something like this. This is done by Guru, who's going to give a talk tomorrow. So what they have is these colloidal particle again, which is decorated with some polymer, right? And then they ice template this. That means they make chains by doing some templating in the system, okay? And then they coat these active particles with platinum. Okay, so now difference between this and the genus chain that I showed you before is in the genus you have a well-defined platinum region and a well-defined silica region. In this case, even though it's platinum coated on silica, there's no well-defined platinum region. Okay, you have platinum everywhere on the colloid, okay, which undertakes this kind of reactions, okay, and by virtue of these reactions, you see that when you have activity, okay, by increasing the rigidity of these, so they have small polymer itself, small chain itself, and by changing the rigidity, they can make it more and more active. Enhanced diffusion is what they show here. But this is slightly different from what is there in the janus chain, right? And therefore the behavior will be very different. Janus chain, for example, shows a very directed motion. Okay, there the particle can propel in a particular direction for a long time. In this case, you will only see the diffusive motion because there is no directionality, but the diffusion is enhanced because of these activities which is happening due to the presence of platinum particle. Okay, so this is another kind of active particles. Okay, so what I'm trying to emphasize by doing this is, again, within the regime of active polymers, there are different types of active polymers that are there, and therefore different types of models are required to understand those polymers. Okay, yeah. Okay, no, in this, the previous case, In this case, yeah. So magnetic field is making them assemble, right? And how strong they will be assembled to each other, right? That will decide the motion, right? If they are loosely bound, then the motion will not be so strong, right? Yeah, yeah. So that's the packing of the how compact they are that will decide the that's what the magnetic field is deciding towards the direction of the magnetic field. Activity will also influence them. Of activity and magnetic field, I don't know, I don't remember the discussion that they had in this paper. Maybe we can go back to this paper and see this. I don't remember that discussion right now in my head. Okay, so yeah, I'll now quickly go through the previous work. Okay, so this is another model system which people use to understand, talk about active polymer, where this is nothing but the worm that we see the day-to-day life, right? Especially in the monsoon season. So these are the worms, okay? And they show that in this paper, they show that if you take the worm, which is like a model for the active polymer, right? Depending on the activity of the worm, okay, they will show this kind of motion. Here what they are plotting is the fluctuation in the end-to-end distance. So if you have more activity, which is this red curve, you will see there's more fluctuation to the end-to-end distance. That means it's showing a lot of shape fluctuations, starting from a straight state to a buckled state and then going back to the straight state and so on and so forth. But whereas if you reduce the activity, this fluctuation on the end-to-end distance becomes less. That means it does this kind of opening and closing much more slowly. Okay, and just for fun, so how they reduce the activity in this case is giving some alcohol to the worm. So even it's very clear that alcohol reduces the activity even in case of worm. So definitely true for humans. Okay, the other model for active polymers, which is there. I mean, other way where you can think of that these active polymers are present is inside our body, right? We have chromatin, right, inside the cell, right? Chromatin is modeled as active polymers thoroughly now in literature, right? This is one such paper, recent paper which I'm talking about, but now the people have started talking about the active polymer models for the chromatin, where where they talk about these heterochromatin and euchromatin. So heterochromatin and euchromatin are present on the chromatin itself, right? Heterochromatin is a part which is closed and which is inactive or less active, I should say. And then euchromatin is a part which is open along the chromatin backbone and which has a lot of activity inside that. And the way these people have modeled it by having larger magnitude of active forces on the euchromatin part and smaller active forces on the heterochromatin part. And then they show that this correlated active force, which is there on the euchromatin part, gives rise to an effective attraction between the polymer segment and therefore the formation of these compartments, right? So there are compartments present in the chromatin, right, which can be modeled using these active forces on a difference in active forces on different regime. Okay. So that was about the experiments which are available, plethora of experiments which are available on the active polymer site. Now there are different types of models also which are available on the active polymer sites. Now this is one such model which is called active Brownian polymer model. Okay, as the name itself suggests, these are the polymers where the activity is Brownian. Okay, and one can think of these magnetic particle in the active bacteria system as the experimental motivation for this kind of model. So what they have in this model is they have either you have a passive polymer with the active medium around it, which gives a kick like these red particles, or you can think of the polymer itself, okay, have each of these monomer having some active Brownian forces. Okay, and both of these kind of model, which falls under the category of ABPO, have shown have shown this kind of non-monotonic behavior in terms of the end-to-end distance calculation. So what you see as a function of end-to-end distance as a function of Peclet number in both these cases, you see that it decreases and it increases. That means when the activity, which is defined by the Peclet number, right, if the activity is low, the polymer tries to collapse, right, or shrink, right, which is shown by these dip in RE. But when the activity is high, okay, the polymer tries to elongate, right? And they explain also, especially in both these cases, they explain in terms of having the curvatures when the activity is small. For example, in this case, the bacteria or whatever, these active particles will make the curvature, which will shrink the polymer, right? But if the activity is very large, these particles is going to slide on these filament, which is going to open this up and therefore will go to a more open state. It's opposite to the, yeah, right. Okay, because these are different system. Warm is a system where you will have some sort of a polar activity. This is a active Brownian type of activity. Yeah.
So how will the activity of the polymer chain change if you let's say these are springs, right? The
Yeah, both of these are strings.
if you
Yeah.
change the stiffness of the springs,
Stiffness of the string, if you change, you mean the flexible or semi-flexible, you mean? Okay, it will change a lot. Yeah, things will become very different once you have the semi-flexible. So this dip that we have, if you have a very small semi-flexibility, you will still see the dip, but if you make it rigid, this kind of dips might go away because the curvature which is being formed, right, by these kicks will be reduced and therefore these dips will also be reduced. Okay. Okay.
I have a question.
Yeah.
So, with the worm case, wouldn't you expect that there is more persistent motion for the worms in
When
case of
you
high activity?
in case of high
Yeah.
activity, yeah, so okay, so in case of worm, the single worm which they have studied in this particular paper, they just talked about the fluctuation of end to end. Okay, so what they have shown is when you have activity, it just does more of this fluctuation. Okay, but if you have less activity, that fluctuation is reduced. They have not really talked about in this paper the directional motion. Okay, therefore I will not be able to comment, but what they show, this kind of shape fluctuation is more with the activity. The main aim of that paper was not really single polymer, single worm. It was actually the rheology of the worm which they were interested in. Okay, and then they show that when you have active worms, okay, as opposed to a passive worm or less active worm, the shear thinning which is observed in the classic polymer cases, that shear thinning effect reduces with that activity. But that was the main aim of that paper. But since I was just talking about the single one, that's why I just talked to you about the single. Okay, so this is another filament, active filament. Here you see a different types of activity. These are called alignment activity. Wischick model, some of you might have heard about, right? What happens in Wischick model is you tend to align with your neighbors, right? So these are the polymers again. This work is done by Subir's group in JNC. So what the monomers do is they tend to align with their neighbors, just like Wischick model. And what you see is a very different behavior here in terms of Rg as you change the Fa. Fa is the activity strength in this case. So what you see is when the activity strength is, there is no activity, okay, the Rg with radius of gyration follows certain behavior. When you increase the activity, it decreases faster, that means it collapses faster, okay. Whereas if you increase the activity further, if you keep on increasing the activity, it's not that the collapse is going to be faster and faster. In fact, after a certain activity, it starts to decrease less. I mean, it takes more time to collapse. Finally, it will collapse, but it will take more time to collapse. So that means there's a non-monotonic behavior that they have observed for this coarsening time. If you increase the rigidity for this particular model of the active alignment, okay? And the reason that they say is when you have very high activity, okay, what happens is these kind of dumbbell sort of structures form where this particular hand tries to go in one direction, this hand tries to go in other direction, and therefore they take really large time to collapse, and that's why the collapse time increases, the coarsening time increases drastically. It's a non-monotonic function of activity. Okay, I'll rush through this again. This is again an active model, polymer model, but here the filament is polar filament, unlike the ABPO where things are hitting at every place. It's just like the magnetic particle on the active bacteria. Here, this you can think of more of a Janus chain, okay, where you have polar activity coming in, and then they show a very nice coil to globule transition if you have such activity. Okay, with that, I come to our model of active polymer. Please pay attention to this slide. If you understand this slide, you know the result completely. So in this slide, what we have is a model for chemically active motor. This is very much inspired by this Janus sort of chains that we have. So you see these segments of C and N, these dimers, purple and pink all over. These are the units which imparts activity. These you can think of as the Janus particles. So if I just blow up one of them, okay, what happens on each of these dimer is there's a reaction that happens. So you have A going to B on each of these C purple particles. Okay, right? So now what will happen if you have a reaction going on one of them, then this is going to create a gradient, which is what I am depicting in this figure. You see there is more B particle, more orange means more product particle, and as I go away from C, the product particle decreases. So there is a gradient which is generated by virtue of these dimers sitting along this monomers. So all these dimers, all these Janus, if you want to think about, are giving this kind of chemical field to the system, right? So each of them, if I just consider one of them, what will happen? By virtue of this, because of this gradient, it is going to show a propulsion behavior, propulsive behavior. This is what I show in here. So there is a flow which is generated, and this shows something some sort of a pusher behavior. Okay, there is a flow which is generated around this flow of the solvent, okay, which is coming from these A and B product and fuel particle, which we have explicit in our model, and this flow is something which is driving these dimers in a particular direction. Now, when I have multiple of them, yeah. Is this a model of any real system? You can think of this as a model of the Janus chain. It's just okay. But the Janus chain that we have there is more rigid. What I am talking about right now is flexible. I'll talk about the rigid also if the time permits, and there we show a very directed motion. But flexible itself is pretty rich. Okay, so when we have something like this, many of these dimers sitting, it will not happen that this C is going to influence only this N and therefore only going to give a polar force. No. What is going to happen in this case is this is creating a gradient, this is creating a gradient, this is creating a gradient, and all the N's, right, is going to sense all the gradient, right? So there are two forces that is coming into picture now. A local force, right, because of this guy only, I mean the N sensing the neighboring C, okay, and then there is these N's which are also sensing the other C's, right? So there is an effective long range interaction on top of the local tangential or polar interaction which are present in this case. Okay, I hope that is very clear to everyone, because you see this C is creating the gradient, N is responding to that gradient. Yeah. Why the passive particles are always right to the this is just one model. Okay, the green one you're saying, right? The pink one. This is just one model. Okay, where I wanted to show that these are the two things which are present. You can flip it, the trajectory changes, the behavior changes, especially in case of flexible one. The semi-flexible one. Flexible one doesn't change much even if you flip it. Okay. These linkers, they're just repeating unit. We took a repeating units of 4. That's why there are linkers at the end. Okay, this also because we thought, okay, we can switch it and put it here and all that, but this is not something which are important. These are just linker passive Bs. The important ones are C and N. C is creating the gradient, N is responding to that. So if I have only one Cn, it will just go like that. But if I have multiple of them, right, this N will also sense the gradient from this coming from this C. So there is a long range interaction on top of the short range interaction.
Mentioned that if you flip this, then behavior will change. Will it change if you flip this?
No, that's what I'm saying. So it will again depend on the sizes. So there are a lot of, so let's say if it moves with only the linker at the front, okay, then the trajectory will be more, I will come to that, more circular. If I have these active particles at the end, then the trajectory would be more straight. Okay, so that kind of minute changes which are going to happen. Boundary effects is going to come, yeah. Okay, so there is this technical reason for that. There is nothing specific. The technical reason is that we wanted this guy, this end, to be slightly far off from the next C. Okay, that's all. That's the technical reason which we have, and that just enhances the motion, the effect, nothing more. Okay, but the idea is that we wanted to have these segments, active segments along the backbone. Okay, and yeah. Okay, so there are three types of particle here. Okay, let me again go back to that. C is chemically active. That means the reaction is happening on C. This A, the product, the fuel going to product is happening only on C. N is chemically passive but phoretically active. That means the phoretic force which is coming because of this gradient is only coming to N. Okay, and L is completely active. Okay, it's not phoretically active also, it's not chemically active also. Okay, that is just linking beats, nothing more. Okay, so the forces are coming from N, which is coming due to the reaction happening on C. And the bottom line, two things. There are, so because in this model you have a local force, right, and you have a global force, right, and then okay. Now these are, if I have made my point clear, then rest of the stuff is something which is very trivial for the next 23 minutes. Okay. Then we have this interactions between the monomer and solvent. By the way, this is explicit solvent simulation, which is very challenging in terms of computational time and all that, but it gives us a lot of ideas about what is flow doing. Therefore, we have chosen this. And you see this epsilon parameter here, which is the effective repulsion between A and B. Okay, and we choose, because N is something which is phoretically active, so we choose A and B to be different on N. Okay, even if you choose that to be different on C and L and all that, doesn't matter. Okay, what you have to keep in mind, as long as they are different, the N particle which is sitting there is able to sense the gradient and therefore give a force, right? So in order to have activity, which I have defined by this activity number, right, I need to have a difference of A and epsilon A and epsilon B. Epsilon A and epsilon B are the parameters that affects the interaction between A to N and B to N. Okay, forget about C and L right now. Those are not really important here because we are just talking about phoresis, which is coming from N. I hope I have made my point clear here. Yeah. So thermal phoresis, so for thermophoresis, I may not have this exact definition of activity parameter. I can tune that. In fact, we are working with somebody in collaboration who's working on thermophoresis, but we are doing something very similar. So you'll have an activity number there also by this thermal gradient, which will be slightly different. The definition of that will be slightly different, but you will have something. Okay, and as I said, we have an explicit solvent model, which is modeled using these MPC dynamics. I think I'll skip that, which is having these kind of details of how the MPC is performed. Let me come to the results of this model. I hope the model is clear to everyone. Okay, now as I said, the activity number is something that we tune by tuning this repulsion parameter, the epsilon parameter in the system. So when epsilon of both of them, A and B, interaction of A with N and interaction of B with N is same, it's like passive polymer, right? And if you have a passive polymer, if you evolve with time, you see that this will remain in the coiled state all the time. Okay, that's the steady state that the passive polymers would like to have. But the moment you have activity switched in, this is another extreme of activity in our model, you see the polymer starts going from a coiled state to a globule state, and you can see a beautiful coil to globule transition, at least by looking at the pictures,
So,
right?
ma'am, I have a question. So does this depends on length of monomer also?
Length of monomer, the size of monomer you mean?
Yeah. Yes.
Okay, so size of monomer comes into picture in terms of deciding the activity. If you have these monomers which are very small, then the thermal fluctuations will win, right? And then the activity, effective activity will be very less. Okay. On the other hand, if you have this monomer which are very big, right, the mass will become very big and then the drag will become very big. So then you have to play between the sizes and get an optimum size which will give you the correct activity. Okay.
So when you make this energy parameter current by a factor of 100,
Mm.
you don't have to worry about computational difficulties.
100 is okay. 100 in this case is okay. But if I want to go beyond that, it becomes challenging for me.
Because then
Okay.
your
Yeah.
integration step has
So yeah, so there is a range of activity which we can achieve by doing that. Yes. Okay. And that's why I'll talk to about another model where I can tweak it in a much more reasonable fashion. Okay. So now I'll go a bit fast because I hope the model is clear and the take-home message is clear. The take-home message is, yeah. The two monomers are simple, pure WCA potential. Pure. So WCA is between monomer, monomer, monomer solvent. Only thing is that I tune the WCA strength for A and B with N. That's all. Rest everything is WCA. Okay. So now you see when I increase the activity, okay, the PE is something which decides the activity. You see a shift of the peak of PRG from the large value to a small value, clearly indicating that there is a continuous collapse which is happening when we increase the activity, which is much more clear when I go for RG versus PE plot. And this is the plot which tells you why this is happening. What is it that the activity is doing to make it collapse? Okay, so as I told you, there are two types of forces here. Okay, one is this global force, long range force, and the long range force in this case, when I have epsilon A and epsilon B different such that PE is more than 1, is something which tries to bring N and C together. Right? So there's a long range interaction that sets in by virtue of the chemical gradient. And on top of that, you have these tangential gradient also, tangential forces also. And tangential forces also is something which is helping the polymers to make a globule. Why? Because if you see the tangential forces, if it is these green arrows here, right? And if you take a vertical and horizontal component, see the vertical component of the tangential force, which is these orange lines, right? That changes sign at every curvature. Whenever there is a curvature, the vertical forces is changing sign. That means that is trying to increase the curvature more and more. And that means that's trying to make it collapse. So both these forces which we have here, that polar force as well as the long range force, is something which is trying to bring the monomers closer to each other, and that leads to collapse of the polymer in case of the active chemical activity.
One general question. So, is there some repulsive forces also usually present in polymers?
See, polymers is completely repulsive force. WCA is what I use for monomer, which is completely repulsive. There is no attractive force from LJ which is coming up. The attraction is coming through the chemical gradient which is set up.
Those are dominant.
Those are dominating, and that's why you see this thing. Because when I showed you the pure PE case, right, which is also shown here, when I look at the Flory exponent, which is how RG scales with time, for a passive case, it will be a coiled-like structure, not a collapsed structure, right? And there the Flory exponent will be is known to have roughly 0.66 is the number, and this is what we observe for repulsive for the passive case. But as we increase the activity, the Flory exponent decreases systematically, clearly showing that you are going from a coil to globule transition as you are increasing the activity. Okay, and this is similar to this behavior. So we see this kind of dependence of Flory activity on this activity number, and a similar dependence is shown in a different paper, the polar active paper which I showed you, which is very different from the mechanism that we are talking about here. So the bottom line is that this despite of the difference in the mechanism of the complete only polar activity, and in our case that our active polymers are showing the similar variation of RG with PE and also of nu, which is the Flory exponent with PE.
Yeah, so with the A and the B, the reactant and the product, with time, the product will keep increasing in your system, and that is the thing that's making it coiled, right?
No, it's not that. Product is keep on increase. It will keep on increasing, but we are also putting the fuel inside it. Okay, there's also a flux. So that means in terms of gradient, we will reach a steady state. Okay, now why they're coming together is because this guy, let's say a C sitting here and N sitting here. Okay, so there is a C and N, okay, which are joined to each other, which are giving these polar forces. Okay, and there's a C and N which is sitting here, right? And this N is also sensing this. Okay, so there is a sensing that is happening. Okay, and that sensing is something which is bringing them close to each other.
But your WCA,
WCA is only for the is there for both monomer and monomer solvent,
yeah,
right? But the strength of WCA is different for A,
for A and
for
B.
such that, okay, so okay, let me go back to this this model again. Okay, so you see this, okay? So if I have an N here, okay, which is sitting in a gradient, okay, forget about which has caused that gradient. Now this N has a different interaction of A and a different interaction of B. That means you see this, around this N at the front you have more B. At the back you have less B, that means more A.
Yeah.
Right? So if I make the A's repulsion more than B repulsion, what will happen? It will have a net force on it.
Yeah.
And that net force will make it move. That is what is happening here. So now when I have this N, right? And now there is a gradient which is coming not only from this C, but all other C's. Right? So that means what? This is actually moving in a net gradient, which is bringing it closer to all the C's.
Okay. Yeah, I was confused about the part of adding more A's into the system, because with time
no, so we give a flux of A all the time.
So it wouldn't really matter on the size of the system.
No, it will not, as long as you have the proper
constant-
boundaries and all settled.
Yeah. Okay, that was my question. Thank you.
Yeah, please go ahead.
If I think like vaguely, first order, if I have a wire and I put tension on it, it will try to taut it, right?
That's the idea, right? That's what you would think, that if I just put a tension, it is going to open up it up, right? But that's not the case all the time, especially if it is flexible, right? It is the flexible stuff, right? So whenever there is this, and this was shown not only by us, this kind of globule formation was also shown by this previous paper, which I showed you, the PRL paper. Okay. So you see this. This orange line, right, which is the vertical component of the polar force, right?
Oh, it's in one direction, that's all.
This green is the tangential force, which can be decomposed into horizontal direction and the vertical direction, right? The purple and the orange. The purple doesn't change sign, but then whenever there is a curvature, right, the orange changes sign. Okay, which brings them. So this is what you are thinking is right when you will have semi-flexible or rigid rod. But the moment you have flexibility coming into picture, that will bring in these kind of curvatures, which we are going to. I'm going to show more. Okay, and those curvatures are something which is going to bring the collapse the polymer further.
I think it should be about the directionality of the tension. Tension is like the opposite direction. Here we have the force in the same direction, right? Because
Okay, yeah, right, yeah. Tension is different and this is different, yeah.
So, and for semi-flexible polymers also,
You see the
bends
buckling.
matter, and these tensions these are forces local. This is not external force. So as a result, locally this is changing, and it can change shapes. Semi-flexible polymers give rise to different kinds of structural transitions. Maybe some of it I'll show tomorrow.
Oh, that's great. Okay, so you'll have that. Okay, so so this is what I was trying to tell you here, that you have, despite of the mechanisms difference, you see a very similar behavior in terms of the Flory exponent and this. Okay, so here I'll go a bit quickly because I have a lot of stuff to tell you. So this is about the dynamical behavior, where we calculate the mean square displacement, and from there we calculate the diffusion coefficient, and we see a very nice increase in the diffusion coefficient, because at the end of the day it will make a globule, right, which will just diffuse around. There is no directionality because it's flexible one, right, and then it has to show the diffusion, and we see that when you increase the activity, the diffusion becomes higher. But the interesting part, a good part, is that for all these activity, the ZIM behavior is intact in the simulation. Why ZIM? Because we have this simulation here, we have this system here, which has explicit solvent, explicit hydrodynamics, and therefore we should go for the ZIM behavior, we should look for the ZIM behavior, and that is what we see here also. In terms of this relaxation time also, what we see is that active polymer relaxes faster. So this is for the passive and this is for the active. The relaxation time is slower for the active. So as the activity is increased, the relaxation the time is decreased, but the scaling is also again the ZIM dynamics is what you see. ZIM dynamics, okay. Okay, so the polymer physics, if you think of, right? So how do you define, how do you understand polymer analytical? There is a well-developed theory which is there in the polymer physics literature, which talks about two types of dynamics, two types of model. One is Rouse model and the other is ZIM model. Rouse model is the one where you talk about just simple polymer, okay, without the effect of flows and all that, and you talk about the this SAW, self-avoiding walks and all that. And then you talk about certain scaling in the sense how RG is going to behave, scale with N, how diffusion coefficient or relaxation time is going to scale as N. That will give you a set of scaling. But if you include the effect of flows, which is hydrodynamic interactions, which is what we have also because we have explicit solvent model, okay, these scalings would change, and that was done by ZIM. That's why it's called ZIM dynamics. Okay, those scalings will change, and that is what the scaling that we see here. Okay, I did not go into the detail of this at all, but just to give you a flavor of what is the difference between Rouse and ZIM. Okay, we can discuss more if you want to know more. Okay, that was about the linear. Okay, what if we make it a ring? Okay, and here also there's nice fascinating stuff that we see here. Okay, so this is a ring where now I told you there's a long range interaction, there's a short range, the local forces, right? Now these long range interaction that I have, I can tune it because in the simulations we can do that. I can tune it to make it attractive, I can tune it to make it repulsive, right? And that tuning, again, this is the same activity number. Instead of PE, now we are talking about AC because PE somehow people got confused with Peclet number, which is not right in our case. So this activity number, this is the passive case. Now if I increase, just like linear polymer, it will show a collapse. But if I decrease that, okay, that activity number, it tries to show the swelling behavior. So this is much more richer now as opposed to the linear polymer. So here we can show a swelling to collapse. Even in linear we can show a swelling, but we have not really worked on that. For this polymer, ring polymer, we thought we will work on the swelling and collapse. Okay, so now here you see the depending on the activity number. So 1 was the limit for the previous case. I was not going below 1. Below 1 is like chemo repulsion. Okay, and above 1 is like chemo attraction. Attraction is leading to the collapse, repulsion is leading to the swelling of the polymer. It's all chemical sensing which is making the polymer chemo attract or chemo repel. The idea is that this is the model that we have, which can very well mimic the chemo attraction as well as the sensing, which likes to tries to bring the polymer close to each other, monomers close to each other, and a repulsion which will try to swell the polymer. Okay, now how do we quantify these collapse? That we do by this something called this percentage deviation that actually tells you this delta Rg is trying to tell you how much for a given activity, how much the structure is deviating from its passive structure. So higher the delta Rg, more is the deviation from its passive structure. Both in case of chemo attraction and repulsion, because this is mod, so it doesn't really talk about attraction or repulsion here. But what we try to show here is if you sit at a particular AC, let's say I'm talking about this large AC, if I increase the number of monomer, okay, which is this inset plot here, right? What happens is that delta Rg increases and then saturates. That means what? That means there's an optimum deviation that it will have from its passive counterpart, okay, which will now not change if you go for a very large limit. For a smaller n, which is smaller ring, right, this percentage deviation is less, right? And that points us to the fact that the ring, the smaller rings that we have, has some sort of a rigidity. Okay, and this is the reason why it is not able to show more and more deviation as the higher rings are going to show. Even though we start with our model has a flexible polymer model, but because of this topology, ring topology, okay, it has some inbuilt rigidity to it. Okay, and that is more clear if I compare the ring and linear together. Okay, so if I compare the ring and linear together, especially the main plot here, which is for the smaller ring, you see the Rg, even though we start with similar Rg, okay, the Rg for ring is much higher, the saturation value of Rg in the steady state is much higher than the linear. And this is just because of the topology of the system. So if I have a ring topology, okay, for a larger size of the ring, it is not going to have any difference. The behavior is going to be very much same as the linear polymer. But if you have a small ring, you have to be careful because the topology imparts a rigidity to the polymer. So even if it is showing some collapse, right, as opposed to the passive counterpart, but how much collapse it is going to show will be very different from the large ring. Okay, this is another interesting thing there where we think that we can correlate our model to the living system. Okay, so what we do here is we take this polymer again, the ring polymer again. Now we change the active site. Okay, we are talking about now only the chemoattractant polymer, okay, which is trying to show the collapse. But what we are changing is the number of active units which are placed on this passive, on this polymer. And we are continuously changing it. And when we change the active units, you see for a very small number of active units, which is 10% of the total number, okay, we see a scaling which is 0.6. 0.6, if you remember, was the scaling for the passive one. So if you have a active sites which is small in numbers, right, it is not going to show the collapse. Very much not so interesting, which is obvious. If you have certain number, beyond certain number, it will start showing the collapse. But what is interesting is this plot, which shows the contact probability between two monomers which are separated by a distance s on the contour. So if I take two monomers which are s distance apart on the contour and ask this question, that what is the probability of contact between two monomers and how does it scale, right, with s, right? For n equal to 7, we see 1 point, s going as 1 point minus 1.8. And if you look at the literature, for a chain with pure self-avoiding walk, okay, that goes as s to the power minus 2, right? But if I increase it to the larger number of active sites, let's say 14, okay, it has a different scaling, which is 1.5. If I increase it further, it has a different scaling as 1. What does it mean? What it is telling you is that as I'm increasing the number of active sites, not only the probability of 2 points which are far apart on the polymer, of them being in contact increases, but the scaling also increases. That means the underlying mechanism of how they are coming in contact with each other has also changed, right? This 1.5, which shows this kind of collapse structure, has a different scaling. But if I increase it to, let's say, 21, which is 25%, it has a completely different scaling. And these scalings are known to come from Gaussian chain, and this scaling is known to come in the literature by something called fractal globule. Now, to motivate you that how this is relevant in terms of living structures, people in chromatin literature has talked about this probability of contact a lot, okay? And it seems that depending on which organism that you talk about, there is this scaling which are different. For example, the yeast, right? In the yeast chromatin, the probability of contact actually goes as 1 s to the power minus 1.5. Whereas if I talk about human chromatin, right, that goes as s to the power minus 1, right? So that means what we are trying to be slightly brave and say that this is a generic model that we have, where depending on the active sites that we choose, we can actually think of employing that model on the actual living systems also. Okay. Okay, so this is something which I'll skip, which is about the comparison between active and passive. This is about the dynamics. We talked about the collapse time, which is before going to the collapse, the time taken, how does the activity decide the collapse time, and what happens to the relaxation time once it goes to a steady collapse state. I'll skip that and come to the last part of my talk. By the way, this work is under revision in soft matter, and we hope to get it published soon. So this is the last part, where what we have tried to do is try to generate, to make a very generic model. Okay, how much time do I have? Okay, there's a lot of questions, 5 more minutes. Okay, so in this case, the previous case was where we had explicit solvent and we had no control. We knew that there are two types of forces which were coming. One is this local tangential force, and then there's a long range force which were coming, which we knew, but we could not get the effect of individuals to find out which is competing, which is having more effect at what range and all that. So here now what we have done is we have this Langevin simulation, simple Langevin simulation, where we have now put these forces separately, because this is now an implicit solvent simulation. Okay, what we wanted to know is what is the effect of the local force and what is the effect of the global force. The local force, local tangential force, is simple, which is force acting on each of these units tangentially. Okay, whereas this global force, which we model as this diffusive phoretic force, comes from a long range chemical field which we have in the system, which we can actually compute this by solving the diffusion equation, and that will give rise to this 1 over r type of long range forces, and that long range force in our case will give a diffusive phoretic force. So this is exactly same as what we were doing with the explicit simulation model, except that now we can actually resolve what is happening due to the local force and what is happening due to the global force. Okay, now in this case we have now two activity numbers, okay, which one comes from the local force and the other one comes from the global force. Okay, and we do the same thing what we had done previously. Okay, we calculate Rg versus N for different combinations of PE and RE. That means the local force and global force. Okay. Now just to tell you that in the MPCD simulation, the previous simulation that we talked about, we could not control these local and global forces a lot. Those values were small. Whereas here, we can now control, we can make it larger, we can see the effect of each other, right? So if you see these kind of cyan color plot, okay, something that we had seen in the MPC simulation, where you see a decrease in Rg when you increase the activity. Here also for certain combination, okay, of PE and RE, PET and PER, which is the tangential and global forces, we do see exactly the same behavior as the MPC, but here now we can be more brave and see if we increase the tangential force, what happens? If I increase the radial force, what happens? Okay. Now this plot looks very confusing because here everything is present. Both the tangential and radial force is present. Therefore, this is very confusing. So what we do is now we plot them separately, keeping the others constant. For example, in the left one, we have only the tangential force. For the right one, we have only the radial force. Okay. One thing which I did not tell you in the previous plot, okay, sorry, that was the effect of rushing. So what you see is that for certain combinations, especially for these, when we increase these two forces, we do see a swelling behavior for the small n, okay, and a collapse behavior for the larger n, in between some complex behavior. Okay, so the question that we now asked is which is the force which is responsible for this swelling, and which is the force which is responsible at the large n size. Okay, and that is what I showed you in the next plot. Okay, so if I now resolve them separately, what you see is if you have a pure long range force that is going to systematically collapse the polymer for all n's, even if it is small, but if it is the tangential local force, local polar force, okay, which gives the swelling of the polymer, okay, which is not present in the linear case. It is only present in the case of ring, right? So in the case of ring, it is this tangential force that gives a swelling, And of course, at the large N regime, you will always have collapse. Now, whether it's coming from this or this is still not clear to us. Okay, but what is clear to us is that non-monotonicity that we see in the previous one, where there's a swelling and then there's a collapse at large N, is coming due to the local force, not due to the global force. The interesting part, the most interesting part of this work, which we found, is when we tried to see which wins over at what N. Okay, for that, this is the purple, the orange one, or orange curve is the one when I have both present, and the red is the one where I have only the tangential present, and this purple is the one where I have only the long range present. And it clearly tells us, so now what I plot in the inset is the percentage deviation of tangential and radial with respect to the orange, which is where we have the combination. So what do you see here? For small n, the red curve is low. That means the deviation from this is low. That means the red curve, which is coming from the tangential, is dominating. So at small n, it's a tangential force which is dominating, and therefore you see a swelling. Interestingly, at large n also, it's the tangential which is dominating. And therefore, the collapse that you see at the large n is not coming due to the radial. Of course, that is also coming into picture. But the actual, the main reason, the main force which is making it collapse is the tangential force. Radial is also there, but the tangential is more competing. The radial is only competing or winning only at the intermediate n. Okay, so this plot is telling us which force prevails at which n. Okay, now it's very counterintuitive that why tangential is going to prevail at large n. Okay, because I always thought, when I saw this result, I thought that radial is going to dominate at large n, and smaller n, the tangential is going to dominate. This result that why tangential is dominating at large n was very counterintuitive to me. And to understand that, we've plotted this bond correlation function, okay, for this large n. What we saw is when you have these tangential activities, you see a dip on this bond correlation function for a small value of s. What does this dip indicate? This dip indicates a hairpin-like of structure in the system. Okay, so when you have these tangential forces, these polar forces, this is going to give these hairpin-like structures even for large n, and those hairpin-like structures, along with these other forces which are there, okay, even for the pure this thing, is going to give you a collapsed state, which is not present when you have the pure radial gradient. Okay, so that's the take-home message. I think I have overshot my time. So with this, so we have also quantified the role of activity, both these activities on the relaxation time. Okay, I will skip this part and come to the conclusion. Okay, so what we have done is we have a model, a very generic model for chemically active polymer. We started with everything implicit, then we have now split it. We now know what is the reason, which is prevailing at what n, and so on and so forth. I did not show you the result, but we have also the results on semi-flexible polymer, which we show that it can be a carrier for a cargo transport. Okay, the work here, whatever I have presented, these part I have not presented. These last three, whatever I have presented is from these three work, which is mostly done by Namita and Sandeep. Very bright student, brilliant student in the lab. So with that, I would wrap up. I hope I did not bore you enough. Morning talk. Thank you so much.
Question?
You showed the exponent s like -1, -1.5, and -1.84. So does that have anything to do with the ratio of the number of active, sites and the number of linkers, like the size of the chain?
Okay, so that one, okay, that's a very good question, because the size of the chain we are limited, because that calculation that we have done is only for the case where we have explicit solvent. Maximum size that we can go to is 100 monomers. Okay, but if I increase that further, okay, what will happen? I don't have the answer to that. if I reduce it, okay, I see a similar behavior. But more interesting question would be what will happen if I increase it, because things will change, things might change there, right? And that's what we will carry out now when we have this Langevin simulations where we can go for larger systems. So what Surbi is trying to do is she's trying to have a one-to-one mapping between this MPCD and Langevin, where we can actually say that, okay, this is what we see, this is the system that we see in MPCD, and this is exactly the system that we have in Langevin. The work that I have shown you is slightly decoupled. We are trying to couple them together, and then we will have the better answer for your question.
Why the local tangential force is constant? Is the concentration of B is not changing?
Okay, in the steady state, if I talk about a single diamond, okay, in the steady state, the gradient would be similar. It will not change much because there's always production of B, okay, and that B is diffusing with a constant rate. So in the steady state, around a diamond, I'll have the similar concentration gradient, and therefore the forces would be also similar. It will not deviate a lot.
So here you are not using the dynamics for the A and B particle.
I am using a dynamics for A and B particle. This is called multi-particle collision dynamics, where I did not explain that, but they're also moving with certain diffusion coefficient.
Okay.
Okay, but that's constant, right?
Okay.
So that will, in the steady state, it will set up a same gradient, and then the forces would be same.
So I was wondering, is it possible to look at the configurational entropy of such system and look at the entropic dynamics?
We can do that. I have not really thought about it. Maybe we can talk and suggest what we can do here. We can do that definitely.
Can you explain more on this hairpin-like structures, how they are formed and the dip in the...
Okay, so the hairpin-like structure, as I just told you in the beginning, right? If you have the polar activity, right? Now, polar activity is something whenever you will have is trying to give curvature, okay? And that curvature, so when it is changing sign, which I showed you, the horizontal and the vertical component, that gives rise to the curvature in the system. And those are the curvature which brings in this kind of hairpin-like structures, okay? And that dip is coming in bond correlation is because you have this kind of hair-like structure. Okay, maybe I can just take 2 more minutes and explain this a bit more. Okay, so what you have is the bond correlation function here, right? So how the bonds are related to each other. So you see a region here. So if I look at just the passive case, which is the black line, it just shows that the bond is decorrelating and it's completely decorrelated at large L. But for these cases, which is the tangential activity, you see anti-correlation. Okay. Now the anti-correlation will only come when you have a bend like this. Okay, the bond is going like that, and it's going to be a negative sign. So this anti-correlation region that you see is the signature of hairpin-like structure.
Thank you.
So I'm wondering that if this passive polymers also goes from this coil to globule transition if the solid is good or
bad
bad.
solvents.
So the shape of the globule, is it depend on the activity, or the globule will always be spherical?
No, so we see that I have not shown you that we calculate the asphericity, okay, and I don't know whether I have the results here or not. Yeah, at least for the ring I have the result here. Okay, so the shape is something that we can calculate by asphericity. Okay, and we see that when you change the activity, the asphericity decreases. That means it does depend on the activity. Okay. But then what you are talking about is the collapse stage of the passive polymer, which we have not studied here. Okay, now in the collapse, what will be the value? So in order to bring in the collapse in the passive polymer, we have to change the model. With just WCA, you will not see a collapse. You will have to bring in the attractive interactions or change the solvent quality, right? Those things we have not done. So I really don't know what will be the value of asphericity in such case, but one can do that. It's not something very difficult.
No more questions.